Welcome to Week 7 of the Introduction 2020 Faculty Perspective Series. Today's roundup of faculty talks is featuring faculty from the School of Journalism and Communication, the Department of Indigenous Race and Ethnic Studies, the Department of Earth Sciences, the Department of Cinema Studies, and the Department of Biology. In a moment, I will turn the program over to our presenter, Dr. Bill Cresko. Before that, though, I'm going to review a few things that are important to keeping you on track to complete introduction. All students watching this program should make time to submit your reflection of this session as soon as we're done here this afternoon. You can do that at community.uoregon.edu. Your reflections on the individual faculty perspectives programs don't have to be long, but we do want you to share what you've learned. Generally speaking, taking time to reflect in this way is a proven way to improve your retention of material and is a habit of tons of successful students. If you're a student who wants to earn a credit as part of your introduction experience, you need to select all of the faculty perspectives within 2 full tracks, plus 4 additional talks, and submit reflections on all 20. You'll also need to submit a longer capstone reflection on the entirety of the programs that you've watched. Finally, for you students who are watching in Zoom, remember that our faculty members are answering your questions live in the last segment of each of these programs. Students, if you're watching live, you should use the Q&A feature at the bottom of your Zoom screen to ask questions of our presenter here this afternoon. All right, I'm going to stop talking and introduce you to Dr. Bill Cresko, Professor of Biology, who will explore how quickly organisms can adapt to new environments in this week's Innovation and Entrepreneurship track. Welcome, Dr. Cresko.
Great. Thank you, Cora. It's a real pleasure to be here today and to actually welcome our next and newest members of our flock to campus. And very soon we'll be able to flock in person, but for the time being, we'll just do it electronically for a little while. And as Cora mentioned in her introduction, I'm here to tell you a little bit about some of the work that we do in my laboratory and other researchers at the University of Oregon on asking questions about adaptation in natural populations. And you'll see on the slide in front of you that there are pictures of a fish and a lake, and those will become clear in a second why those are there. But let me just start off by saying a little bit more about the laboratory. So what is a laboratory at the University of Oregon or many places? Well, it's a physical space. It's a set of tools and techniques and pieces of equipment, but more than anything else, it's actually the people in the laboratory. So our lab and many labs at the U of O only run on the excitement and the energy of the people who come and work with us on our research. And one of the key things that I think differentiates the University of Oregon from many places is that undergraduates are really a key lifeblood of research in laboratories. So this is just a subset of undergraduates who have worked in our laboratory over the last decade since I came to the University of Oregon. And many of these people have gone on to careers in academics, in government, in the private industry, but all of them played key roles in doing real work and real science in the lab. So my talk today is actually going to highlight some key entrepreneur and innovation that's happened through undergraduate researchers in the lab, many of whom did their honors theses. So this is something to consider, that many of you might be able to come and work in a laboratory, work with a faculty member, graduate students or postdoctoral scholars, to actually not only take courses and get credit, but to do an honors thesis. And so here's a set over the last 4 years of honors college, Clark Honors College thesis students and biology thesis students in my laboratory. So Starla Shambros is still a student in my lab. Jack Poplinski and Emily Niebergall just graduated last year, as did Jared Galloway. And John Crandall, Taylor Wilson graduated a couple years before, and Kate Atworty and Sophie Sichel. And I'd just like to point out that among these students who I'm showing you here, they, together with us, have published a total of over 10 papers, manuscripts, in peer-reviewed journals, and have done really amazing things. And I just give a couple examples of Emily Niebergall, who is one of our 3 Goldwater scholars last year at the University of Oregon, John Crandall, who graduated and is now in his PhD program at the University of Wisconsin Laboratory in Genetics, Taylor Wilson, who wanted to go to vet school and ended up at one of the best in the world, Tufts, and then Kate Atworta, who ended up going to the Cleveland Clinic, one of only 32 students, and now is actually a resident at Harvard at Beth Deaconess after coming as a first-generation student from a Latinx family in New Mexico. So being a researcher in a laboratory allows you to do great things, and more importantly, to really expand your mind to ask amazing questions like, how do organisms adapt to novel conditions? Well, in some cases, organisms adapt to novel conditions by changing immediately their physiology. All of us sweat when it gets warm. We actually then get shiver when it gets cold. But really the questions in my lab have to do with the diversity of life that has to do with genetic changes, or the changes in the DNA that occur in the genomes or the entire compendium of genetic material in organisms. So a fundamental question that's fascinated not only biologists but humans is, how did this diversity of life arise? What are the processes that actually occur via natural selection, changes in local population sizes, but more importantly, what's been happening at the level of DNA sequence? And that question has been very difficult to answer up until just a few years ago. And part of the reason is that, for example, in the genomes of our body, each one of our cells has 3 billion base pairs of DNA. So try to think about if each one of those base pairs was actually a book in a library, that would be 1,000 libraries of Congress. Unbelievable amounts of information to actually sort through and to figure out. So what we in my laboratory and many other researchers do is we focus in on model systems. And those model systems, or groups of organisms that we study, we do so for a variety of reasons. You may have heard talks about or done research or learned about things like fruit flies, which are really good for genetics in the laboratory, or nematode worms that are really workhorses for understanding how neurons in the brain work. The organism we use in the laboratory for a lot of our studies is this small fish called the Threespine stickleback, or Gasterosteus aculeatus. This fish is actually really interesting because it lives in coastal habitats around the northern hemisphere. I'm just showing North America right here at the height of the last glacial maximum. But if you go, for example, when I leave the Redwood ballroom that I'm in now and go outside, if I were to walk across Franklin Boulevard, I could see stickleback swimming around in the mill race right next to Willamette River. In many of these regions of the northern hemisphere that were glaciated 18 to 20,000 years ago, as glaciers receded, new habitats opened up, such as that one that's shown in the lower right-hand corner, and one of the first organisms that invades is a marine form of Threespine stickleback. And what's happened afterwards is an amazing explosion of diversity. So this is just a whole bunch of pictures from a review paper from 20 years ago that just shows pictures of the male nuptial coloration. So these are the colors that the males take on when they're build nests on the bottom of lakes and try to court females to convince them to come into the lakes, or to come into the nest, lay their eggs, and then the males will take care of them. So each one of these pictures shows you pairs of stickleback that exist in closely connected habitats, some in which the males turn bright red, some where they turn jet black, some that get more orangey, some that get blue. And that's just one of the many different characteristics or traits, or the term that biologists use, phenotypes, that changes in this organism, and they happen quite rapidly. So as I mentioned, many of these fish live in habitats that were deglaciated at over the last 13 to 14,000 years. And one of the key changes that's occurred is in the bony armor. The Latin name, Gasterosteus aculeatus, means spine belly bone. So you'll notice that what I'm labeling here as the pelvic structure is modified pelvic fins of the fish. So if we actually think about, say, a salmon or a trout, they'll have pectoral fins and pelvic fins. That's a modified defensive structure. Additionally, the lateral plates down the side are a bony armor, not too different than, say, chainmail for, if you could think of from knights in armor. And these structures have evolved because these small fish get eaten quite a bit by larger fish, or in the bottom, that's a picture of a dragonfly naiad that will actually grab onto the fish and try to eat them. And so it's been shown that those armor structures help these fish survive. But they're very expensive to make energetically. And including they have to eat more, and they have to live in the right habitats that have enough calcium and other minerals, and that comes at the expense of reproduction. So there's always a trade-off in allocation to the creation of those structures and then not creating those and rerouting that energy to reproduction. So work on this organism for years has really laid out its understanding of its ecology, behavioral changes that are really quite striking, differences in morphology, which is for example, the size and shape of the bones. And when I started on this work about 15 years ago, a big question for me was, what's going on inside the genome? What genes are actually changing? What DNA sequences are evolving? And at that time when we started, there really wasn't an ability to answer this question. And so I and one of my colleagues, Eric Johnson, who's also a faculty member in the biology department, were new faculty. We both had just opened our laboratories at the University of Oregon. And at that time in the mid-2000s, there was a revolution occurring in a type of sequencing technology called next-generation sequencing technology. And Eric and I quickly realized that there might be a way for us to go through and harness that sequencing technology to create a new genomic scanning technology that was much faster, cheaper, and more efficient than anything that'd come before. So it's actually, we don't have quite enough time to go through what I'm showing you on the slide here on the upper left-hand corner, but suffice it to say it's a representation of the ability to look at thousands and if not millions of locations in the genome. And down below I present two manuscripts or papers that we published that describe this technology. And I do that because it's really important to note that the two folks on the bottom, Mike Miller and Joe Dunham, who were key first authors on these papers, who helped us, were both undergraduates at the time. So they were actually undergraduate researchers who helped us and others develop this new innovative technology. So what can you actually do with something like this? Well, this actually led us to the ability to go through and ask some really interesting questions. And one of these was a question that I and one of my colleagues, Frank von Hippel, who's in the upper right-hand corner of this slide here, who was at that time a faculty member at the University of Alaska Anchorage. Frank and I are long-term collaborators, and actually ends up that his uncle, Pete von Hippel, is a faculty member at the University of Oregon in chemistry department and a world-famous chemist. We asked this question about a very interesting set of islands in Alaska that we know a lot about, and I'll tell you more about those in a second. And to help us study this, we pulled in a set of researchers, Emily Lessig, who is a graduate student, an undergraduate and then a graduate student at the University of Alaska Anchorage, Mary Sherbick, who is an undergraduate researcher who is also an Alaskan native, first-generation researcher at the University of Alaska, John Crandall, in my laboratory at the U of O, and Taylor Wilson, along with several of our graduate students and postdocs. And this is just one example of a paper in a journal that's called the Proceedings of the National Academy of Science, where we first described the genomic changes, or changes in the DNA, that were occurring in these special populations. So what's so special about them? This is the part that was really exciting to me, is that in contrast to a lot of natural exploration of natural changes in ecology and evolution, where we might not have an exact idea of when the glaciers receded and opened up new habitats, in this case we do. It actually happened on March 27th, 1964. The largest earthquake in the northern hemisphere, the second largest ever recorded, happened in south central Alaska. And so that's a picture of the beginning of the Aleutian Peninsula there. Anchorage would be kind of in the middle of that picture on the left. And then the images on the right are downtown Anchorage, where people went into stores and shops before the earthquake hit. And then when they came out, they realized that their cars were 8 to 10 feet below where they were before. They were in buildings looking out at the second, at a street that before had been 2 floors below and was now a floor above. And this earthquake was so massive. A good way to compare this is the Loma Prieta earthquake that happened in California in the late 1980s was about the earthquake in Alaska was about 1,000 times stronger than that one. So what did it do? Well, it actually did a lot of things. It destroyed houses and knocked down buildings, but it also created new land. almost instantaneously. And there were a series of islands on which this happened. And that new land on which these islands were, the islands on which this new land was created were way out in the Gulf of Alaska. And they were way away from other mainland populations of freshwater forms of stickleback. And the only ones that were out there were oceanic forms. So what I'm showing you on the picture here is images from this island, one island out here called Middleton Island, before and after the earthquake. And interestingly, many of these islands weren't inhabited, but we actually knew a lot about them because of the strategic positioning of these during the Cold War, actually not too far from Russia. So when you're looking at the box on the left that's on the island, the picture of the island in 2008, you can see that it's encompassing parts of what were the old island before the earthquake, and that new terrace that was created afterwards. And if you look closely on that new terrace, you can see all these little ponds. They're not very big, but when Frank and his team started to explore them, he realized that they weren't saltwater, they were freshwater ponds. Now, these aren't spring-fed, but there's a lot of rain in this part of Alaska, so it didn't take very long for these small indentations that were created to be filled up with fresh water. They're intermittently connected to the ocean, so they had access their access to these ponds was available to oceanic forms of stickleback, the ones that I showed you before with the really robust armor. And what had ended up happening is when Frank and his students and we went out to Middleton and other islands in the region, one called Danger, there's another one called Montague, and it was quite a trip. So it's hundreds of miles out there. You have to actually take planes and helicopters, put traps out in the field. And in the upper right-hand corner, you can see just on the hand of one of the undergraduate researchers, pictures of those stickleback. The top two are the oceanic form, and the bottom two looked a lot like freshwater stickleback, despite the fact that there was no documentation of freshwater stickleback on that island before the earthquake, except in one small location. And so we asked the question then, how did this happen? Is this a situation in which that these fish have actually evolved over the last 30 to 40 years? So instead of thinking about evolution happening on the order of millions of years or thousands of years, could it actually be happening over human lifespan? And so what we did is we went through and we did a whole bunch of phenotypic characterization, meaning looking at the traits like how big the eye is, the size of the opercle, which is marked by that number 4 right there, which indicates what type of food that they're eating, how much of that armor they had. And we found that when you examined at the phenotypic level, the stickleback that were in the ocean and in freshwater, they were very similar to the ocean and freshwater divergence that we had seen previously that we thought took thousands of years. So the question was, did these fish actually evolve over 50 years or fewer? To do that, we went through and used that rad genotyping technique that we had developed earlier and scanned through the genomes of over 1,000 fish to look at what was actually going on. So we went to different habitats on 3 different lakes, Danger, Montague, and Middleton, and looked at numerous different populations that only had fish that were low-plated or didn't have much of that armor, completely plated, some that lived in freshwater habitats, some that lived in ocean habitats. And the upshot is that, yes, we could actually confirm genetically that these populations evolved independently at least 7 different times, if not more, on each one of these in total across these islands, and did so in ways in which the genome changed only in parts of the regions of the genome that were important for adaptation to that new, different environment, whereas the rest of the genome didn't change at all. And I'll show you what that looks like across the genome. It's pretty hard to look at the whole genome of stickleback fish because it's 500 million base pairs. But we actually have a representation that will actually help you think about it a little bit. But before we do, I just want to point out that not only did these 3 islands get uplifted, were they uplifted during that earthquake, there were hundreds of other locations where this was likely happening over and over again. So what I'm showing you here, it's a little confusing, but on the left, I'll try to talk you through it. The left side of the image is just those 3 islands again. And the color coding is what we described as different groups of populations that likely evolved independently. And on the right is a circle diagram from the inside out that shows the entire 21 chromosomes of the Stickleback genome, 500 million base pairs, separated into each one of those 21 chromosomes. So just like we have 23 chromosomes, Stickleback have 21 chromosomes. So as you're moving along there, green means there's no difference when you look at the ocean versus one ocean population versus something else. And each row from the inside out is a comparison between a marine or an oceanic stickleback and something else. So the first circle is one marine population on one island and another marine population on another island. So what that means is that you see that everything's green. That makes sense because the fish in the ocean swim back and forth. They interbreed. They actually exchange genes or variants called alleles, and they just move and swim back and forth. So we see very little differences on that inner circle. But as you move outwards, and the second circle from the middle, the third, the fourth, everywhere where you see red, right there, indicates locations where there's much greater difference between the freshwater part of the genome and the ocean part of the genome than occurs in other parts of the genome, like here. So, for example, when you look in chromosomes 14, 15, 16, and 17, as I've circled on this, most of those sections are green or slightly yellow, meaning that they're a little bit different, but not very much. Whereas these parts of the genome, where it's red, means they're very different. They're actually so different that they're about the same level of divergence between the ocean and freshwater stickleback as you would find sometimes between different species. And again, this has happened in 45 to 50 years. So to finish up on this slide, each of those rings from two, the second ring, the third ring, the fourth ring, the fifth ring, and the sixth ring are different freshwater populations that evolved in 50 years on 3 different islands that are dozens of miles away from each other, separated by the ocean, and the freshwater fish can't survive in the ocean. So this must have happened independently, but you'll notice it's not random with respect to the genome. In one population where you see a change, you also see that change in another population. For example, in linkage group 4, which is approximately around 230 on the, if this were a clock, you can see that those same regions over and over again change. Linkage group 7, which is around, it's in the lower right-hand side, same thing. Linkage group 11 and 21, which is right up at the top, which looks like around midnight. Interestingly, previously we had shown that these are the places in the genome where genetic variants were segregating, meaning they were in the population that were linked to changes in the phenotype that are the divergence between ocean and freshwater. So that was really cool, but we want to know why it was the same regions over and over again. And what John Crandall, for his undergraduate thesis, did is he said, you know, I bet if we were to take oceanic and freshwater stickleback, including freshwater stickleback that we combined with a marine stickleback and made a hybrid and then crossed those back, what we'd actually find is that there would be some genetic incompatibilities that would lead to a reduction in the recombination, what is called the recombination rate, or the turning over and the connections of the genes, making it easier for cassettes of the genome to actually adapt in freshwater. So John made these crosses, and he did it in the laboratory, and that's another benefit of this organism is we can go out into the field and bring them into the laboratory. And we've actually established laboratory lines of stickleback from all over Oregon now that we use in the laboratory to do crosses and actually genetically modify their genomes. And when he did that, what he actually found was something quite amazing. So instead of showing all 21 of the linkage groups or the chromosomes of stickleback, we're just focusing in on one of these, linkage group 21. So I'm just going to go back a couple slides, and linkage group 21 is that one that's right at the top, almost at midnight on this clock. And you can see those red lines that occur repeatedly on each one of those. So when we look at that, those red lines now represent that middle part of the chromosome. And what's interesting about that middle part of the chromosome there, where it's very red as compared to the blue regions, that's the greatest level of divergence. So that figure right there actually represents FST, which is a measure of divergence within as compared to between. So when you're closer to 0, there's very little difference. When you're closer to 1, there's a lot of divergence there. And just to put that into perspective, when we think of all the human, global human genetic diversity, the FST values are almost all just around 0. Nowhere close to anything, as you see, as diverse as what's here. So what John did is he actually made what are called genetic maps. And those genetic maps then are using linked markers that segregate different ways depending upon which genomes are combined together. So if we think about making different hybrids of horses and donkeys to create mules, you have different changes in how the parts of the genome move with respect to one another. And what he found is that in the crosses where there were just the freshwater genome, represented by that blue representation of the stickleback, right under the sign that says, or the label that says genetic maps, they actually look pretty much the same. They actually go 1-to-1. And then when you looked at the red stickleback, which is just the ocean and ocean genomes together, pretty much the same distance, but it was flipped. And that was a key thing because, that actually indicates that this is something called an inversion, where in one part of one genome of an organism, it's in one orientation, but in this case, 2 million base pairs of DNA had been flipped over. And what John found is then when you bring together those two genomes, that recombination rate over that 2 million base pairs goes from a significant amount to nothing. So that's what the bottom row is showing, how all of those genetic markers, those rad genetic markers, go to 0. And so this research was really exciting to us, and it was exciting to a lot of people in the world. And John actually is a co-author just a few months ago on a paper in the journal Genetics, which is one of the premier journals in the field of genetics. So that wasn't, even though I only showed you linkage group 21 or chromosome 21, what it actually ends up is that the physical genome, or the complete number of base pairs, the 500 million base pairs, of those regions that show the greatest amount of divergence, that accounts for about 20% of the genetic map. But all of those regions across the, 20% of the physical map, or the entire DNA sequence, all of those regions showed the same pattern. All of them, when you actually had the hybrid or combination of fish together, they shrunk down, meaning that even though those parts of the genome that needed to be moved from ocean into fresh water and had to do so very quickly, it wasn't too hard to do once hybridization between ocean and freshwater forms started to happen, because they actually became smaller units that actually could be changed in frequency quite quickly. And I didn't have time to tell you about it today, but one of the folks that I mentioned earlier on in the slide, Taylor Wilson, did very similar work on Oregon populations and found similar patterns. And then Jared Galloway actually was a computer science undergraduate major who worked with faculty member Peter Ralph and myself, and actually developed a new modeling technology and approach where he was able to go through and reconstruct what was happening in the wild that we saw from our data in silico, meaning in computer programs. And that paper was just published a few months ago as well, too. So in summary, to get at this question of how do organisms adapt to novel conditions, it's going to take many, many years of research over many laboratories for long periods of time. I don't want to pretend like the answers that I'm giving here today are the complete answers, even just for Stickleback, which is just one of millions of different organisms, and it's only for questions at the genetic level, let alone these diversity of questions about at the ecological and behavioral levels that are so fascinating. But the fun thing is, you all have your lives ahead of you. If this is something that's interesting to you, not only I, but many others at the University of Oregon have really fundamental, deep questions about this, whether it's Kirsten Sterner and Nelson Ting's research in the Department of Anthropology on non-human primates and the evolution of zoonotic viruses. Well, like SARS-CoV-2, the virus that causes COVID-19. Or if you're interested in studying how host and microbes interact with one another and evolve, you might want to check out research from Karen Gilliman or Brendan Bohannon or Judith Eisen, who's very interested in how that the microbes interact with development to affect how organisms actually behave. But all of these things are available to you and at the University of Oregon. And even if we can't be in person quite yet, a lot of this work can actually be done at a distance. So don't hesitate, don't be afraid, just reach out, because I spend a lot of days and a lot of my time in boring meetings. One of the things I love to do is get an email from somebody who's interested in the work that we or others are going to do. That's what we love to do, and we'd love to hear from you. So I'm looking forward to seeing you in person to help answer some of these questions. And so just to summarize the talk from today, some organisms can rapidly move and adapt to different habitats, like we showed for Stickleback. But what we found is that to do so, they have what's called pre-existing genetic variation or standing genetic variation that exists in ocean populations that can be rapidly moved. So the ability to do this in organisms like Stickleback might help some species, for example, respond to things like global climate change. But it varies from organism to organism. And it really matters whether the organism like the Stickleback, where there's hundreds of thousands or millions of them, have standing genetic variation, or whether we're talking about a very small population that's confined to a region on, say, the top of a mountain, where it really isn't much genetic diversity and really not many places to go. So those organisms may not be able to adapt. And these new technologies, like the one that Eric Johnson, myself, and our students have developed, can help us read the genome like never before. And in our genomes are written, and when I say we, I mean all of organisms on the planet, the history of life. It's amazing to think about the fact that all DNA that exists in our bodies right now can be traced backwards in time to the same piece of DNA that exists in one of our ancestors. They're different people, but they can all be traced back through time. And all organisms that you see around you have genomes that can be read in this way to answer amazing questions. So the stuff of life is written in genomes. So with that, I just want to thank you for listening. It was really fun to come and talk science today. And I want to acknowledge, this is a few years old, some of the members of the laboratory. I chose this picture because many of these are undergraduate students, some of whom I mentioned, others who are actually off in the world. For example, Kat Milligan-Myrie in the upper right-hand corner there was a postdoctoral scholar with Karen Gilliman, myself, and is now a faculty member at the University of Connecticut, Julian Katchen at the University of Illinois. Many of the undergraduate students, actually makes me a little sad to look through this picture and know that they've all grown up and gone off many of them have gone off in the world. So we're ready for the next group of ducks to come in and do work in our laboratory. And special thanks to my mentors. When I was an undergraduate, Neil Shubin was the first person who took me under his wing and taught me all about the fascination of biology. And my PhD advisors, Susan Foster, John Baker, and at the University of Oregon, special mentors, John Postlewaite and Chuck Kimmel. So I want to thank them. And with that, I'm happy to take questions. Thanks.
Thank you so much. We do have questions for you. And actually, I wanted to start off with, because I can ask the questions first. I was thinking, as I listened to you talk about Jared, who's a computer science major, working with you, are there other students who aren't biology majors or the traditional majors that you might find in your lab? Like, how many other kinds of majors have you mentored or supervised within the work that you do?
Yeah, one of the fascinating things to me is how the U of O, in being a comprehensive research and deep liberal arts education institution, is it actually, many of the students are coming from diverse degrees and majors. So for example, Kate Atuarta had dual degrees in French and biology as well too. Computer science, I'm looking up, it's math, anthropology, Russian literature, a variety of different backgrounds. So no, doing work in a laboratory and being interested in research, not only is it not that it should be confined, I actually find that to natural sciences, but I actually find that the undergraduate researchers in the laboratory who have these diverse experiences and interest and degrees actually can put pieces together in very different ways than well, those of us who have been around for a little bit longer.
Thank you for that. Ryan wants to know what determines how fast an organism can evolve, and are there ways to help humans evolve and adapt to new environments faster?
Yeah, so that's actually a really important question that it's not answered yet. So what is it that allows some organisms to adapt quickly and other organisms not? We do have some hints about from research like mine and in our laboratories and others, which it has to do with genetic variation that exist already in populations. So if that genetic variation can be utilized, then changes can happen more quickly. Another good example is domesticated dogs. So if we think about, I myself have a Siberian Husky at home, and Siberian Huskies look kind of like wolves, but not exactly, but Chihuahuas don't, nor do Great Danes. And so most of those differences among breeds of dog that have evolved under artificial selection happen because of the use of existing genetic variation and not new mutations that actually occurred. So in terms of human evolution, humans have evolved and are evolving and will continue to evolve. But frankly, many of the changes that we're facing on the planet are things that, in terms of responses, have to do with not evolution, but with using our resources wisely, actually collaborating, working with one another, and sort of doing the things that we know that we need to do. So responding to the changes that are actually happening on the planet with respect to humans, I first would look to all the other disciplines and degrees that are out there that before I would think about human evolution as a part of the solution.
Thank you for that. Another question that we have here, were your findings pardon me, I'm going to start over. Was your finding of such genetic change surprising to you? Has your research impacted other research done by different folks?
Yeah. So it was surprising in some ways. One thing that wasn't surprising was that we predicted that it really had to be existing genetic variation that was being repurposed. What was surprising about that is that we predicted, and I predicted, that it would be a lot more of the genome, that there would be a lot more complex patterns of different genes, different alleles or genetic variants, but it ended up being kind of more of a simpler pattern, fewer of those genes being utilized over and over again. And in some ways, that actually links to the previous question, because it may mean that one of the interesting conundrums is, even though there are millions of stickleback populations around the world, there aren't that many species of stickleback. There actually are only a few of them. They haven't radiated into different species like, say, the 2,000 or so different African cichlid fishes. So it may be that the simple genetic architecture or the genetic combination allows rapid evolution so far, but then makes it difficult to move much further. So our research and much of the research of the undergraduates who have worked in the laboratory have actually impacted researchers around the world. And if you look at the papers, they end up getting cited, meaning other researchers from everywhere around the world look at those and use them as a guide for the work that they're doing, but also conceptually too, to think about how fast evolution can happen. I'd say that I think we've played a role in that, which has been nice.
Thank you. Okay, so this is a great one. Are there some specific courses in your department you'd recommend for non-science majors to improve our science literacy?
Yeah, so in biology, there are a number of intro courses at the 100 level that are really exciting and are fun courses to take. And so I can think of several of them off the top of my head right now. So there's an intro to human genetics that's offered actually by the head of our department, Bruce Bowerman. That's a really fun course to take. There's courses that are introductions to ecology, introductions to evolution. And one of them is Professor Alan Kelly, he's one of our star faculty at the U of O, in particular with teaching a number of different classes that have to do with cancer biology. So he teaches a course at the 100 level, an introduction to cancer biology, which I think would be really fascinating for students to take. And similar to our department, there are many other departments that offer such courses. For example, human physiology, chemistry, but anthropology as well too, geography, earth sciences, many different departments. And a new one that is just starting this fall that one of my other roles is the executive director of the Data Science Initiative. We have a new degree, an undergraduate degree in data science. And this coming winter, for the first time, we will be teaching the first of 2 courses that's called the Introduction to Data Science. So the use of data and big data to answer questions. And that one is really geared towards students who are interested in just the basics of data science. So that might be a good one to take.
I was going to ask you about that new role of yours. How did that evolve, and why is a biology professor helping shape the data science degree program at the U of O?
Yeah, that's a really good question, because data science can be described and thought as different ways by different people, but there is a core there, and that's that in general folks think about data science as being the harnessing of large amounts of data, or big data. And by that we mean there's a lot of it, it's really variable, it's coming at you really fast, and you're not sure which data to trust. And then the other aspect of it is the use of advanced technology for analyzing it. That might be statistical tests or machine learning approaches in computer science. So that's 2 of the key things. The third key thing about it is that in contrast to computer science or applied mathematics, where those fields are really focused on the aspects of computer science and applied math almost completely, the questions in data science, or the focus, is almost always on applications to some other domain, like in chemistry or in linguistics, where people are really interested in studying entire tomes for corpus linguistics, or in biology, when you create a new genomic technology and realize that you've just created more genetic data than existed before 1990, and realize you didn't have any idea how to analyze it, and you really needed to work with computer scientists and become one yourself to figure out, data scientists, to actually develop new tools on the computational side. And so a big part of our research in our laboratory has actually been developing the data analytic tools for the technologies that are developed. So even things like brain scans from an fMRI in neuroscience generate terabytes, gigabytes and terabytes of data that need to be analyzed. So data science exists across the spectrum of applications in business, in law, in the natural sciences, the social sciences, and humanities. It can be a really key degree all across the board, and that degree that we've developed actually has domain applications in all of those areas.
Thank you for that. Okay, so last question for you. What can new students do in their first year to prepare themselves for roles in research labs like yours?
Yeah, I'll give an example of how I started when I was an undergraduate, because I didn't know what to do. I had no idea how to actually get started. I started asking around and asking my friends, and I found one of them said, well, I got this job and I've started to clean fly vials, which are these little vials that fruit flies live in. And I said, well, that sounds good. I'll get paid and I'll start learning more about that. And when I was in high school, I really knew I liked biology, but I didn't really know what it meant to actually be in a laboratory. And honestly, that's a great way to actually get going in a laboratory, is to just start helping and volunteering and just learning about what happens in that lab, what happens in that group. You can start going to the lab meetings, and very quickly you'll start seeing places where you can move from doing more of the basic work to helping a little bit more, to then getting connected with a mentor in that laboratory. And in my case, And that's exactly what happened after my freshman year and my sophomore year. Then I quickly started developing my own research questions, and in many ways didn't even think about it because it was just, I was part of that lab. And so many of the laboratories and research groups at the University of Oregon expect that students who are coming in their first or second years are going to be starting at one level and then we'll move up. And so, for example, in my lab, one of the first places we get started is, well, somebody's got to feed the fish. How do you feel like about feeding the fish today? Now that you've fed the fish, let's start talking about fish biology. Now let's move on to the next thing. So don't hesitate, ask, and don't feel like you need this really high level of expertise because nobody has it. None of us do. You always have to start somewhere. So just reach out and ask.
Thank you. That's a terrific place to end on, and that's all the time that we have for questions. To you students, please log on to community.uoregon.edu to complete your reflection of this program. Also worth noting for students, we're at a critical point at the end of the summer where you're going to begin to get more and more information from various departments on campus. It's really important that you continue to check your @UOregon email account and read those emails with attention. All of the emails that we're sending you in the coming days and weeks will have a call to action for you, and taking action on these emails will be critical to your readiness for the fall. For questions about this or any other introduction program, you can go to our website, orientation.uoregon.edu. You can email us your questions at orientation@uoregon.edu, or you can text us those questions at 541-346-1159. Thank you to you students who are watching live with us for your time today, and to you, Dr. Cresko, thank you so much for helping our new ducks get ready for fall.